For most of the 20th century, the story of how humans first reached the Americas seemed reasonably settled. A land bridge, Beringia, the exposed continental shelf connecting Siberia to Alaska during the last glacial maximum, allowed Asian hunter-gatherers to walk into a continent no human had ever seen. They arrived, spread south rapidly, and became the ancestors of all Native American peoples. The Clovis culture, identified from distinctive fluted spear points found across North America, was their archaeological signature. The timing was roughly 13,000 years ago. The source population was Siberian. The model was elegant. Then, the genomes started arriving, not the genomes of living populations alone, but the ancient DNA extracted from the teeth and bones of people who had been dead for 10, 12, 15,000 years. And one finding, buried in the data from a study of Amazonian tribes in the remote interior of Brazil, was so unexpected that the researchers who found it initially assumed they had made an error. They had not made an error. They had found a second founding population of the Americas, one that had entered the continent separately, left almost no archaeological trace, and survived only in the genomes of a handful of remote groups living in the Amazon rainforest. The first piece of evidence comes from a 2015 study published in Nature by Pontus Skoglund and David Reich at Harvard Medical School, working alongside Brazilian geneticists who had spent years collecting DNA samples from indigenous communities in the Amazon basin. The team was conducting a large-scale survey of Native American genetic diversity, comparing modern and ancient genomes to build a more accurate picture of how the Americas were initially peopled. The analysis was systematic, and the expectations were straightforward. The genomes of Native American peoples should trace back to a single ancestral source population in Northeast Asia, consistent with the Beringia crossing model. When they ran the analysis on samples from two Amazonian groups, the Suru and Cariciana peoples of the Brazilian interior, the results did not fit. These populations carried a genomic signal that bore no resemblance to any known Native American or Northeast Asian population. Instead, their ancestry, in a statistically significant fraction, was most closely related to indigenous Australian, Melanesian, and Andamanese populations, people from the other side of the Pacific, separated from South America by the world's largest ocean, had contributed detectable ancestry to groups living in the heart of the Amazon rainforest. Skoglund and Reich named this ancestral ghost population Yai the Y from Yai Paikura, meaning ancestor in the Tupi language spoken by groups related to those carrying this signal. They proposed that population Y represented a second migration into the Americas, distinct from, and later than the founding migration that gave rise to the majority of Native American ancestry. This second wave had entered the continent by a route unknown, at a time unknown, carrying ancestry that connected them genomically to populations from Oceania and South Asia, rather than Northeast Asia. The second piece of evidence comes from an independent line of research examining ancient skeletal remains from South America and their unusually variable cranial morphology. Paleoanthropologists had long noticed that the oldest skeletal material from the Americas, skulls recovered from sites in Brazil, Chile, and Mexico dating to more than 10,000 years ago, showed cranial shapes markedly different from later Native American populations. The most famous of these was Luzia, a female skull recovered from the Lagoa Santa cave complex in the state of Minas Gerais, Brazil, dated to between 11,000 and 12,000 years ago. Her facial structure did not resemble East Asian or Native American morphology. It resembled Australasian populations. For decades, this observation was controversial and contested. Some researchers argued it reflected normal variation within a single founding population. Others proposed a separate migration. Without genomic data, the debate could not be resolved. The ancient DNA recovered from Lagoa Santa skeletons, published in 2019 by a team, including Skoglund and Brazilian colleagues, did not fully settle the controversy, but deepened it productively. The Lagoa Santa individuals carried the population Y genomic signal at elevated levels, far higher than modern Amazonian groups. This suggested that the ancient people of Lagoa Santa belonged to, or were closely related to, the population Y lineage, and that over the subsequent millennia, interbreeding with the mainstream ancestral Native American population had diluted that signal in their modern descendants. The ancient signal was strongest at the beginning, It had been fading ever since. The third piece of evidence concerns the timing and route of population-wise entry into the Americas. And here, the genomic data forces an uncomfortable confrontation with what the archaeological record does and does not show. If population Y entered the Americas separately from the main founding population, it must have done so either through Beringia, the same land bridge route, but at a different time, or through a coastal maritime route that bypassed the northern ice sheets entirely. Some researchers have proposed a kelp highway model, in which people moved along the Pacific coast by boat, hugging the coastline from Asia southward, and eventually reaching South America without leaving the kind of interior archaeological sites that land-based migration would produce. The genomic dating of the population Y divergence suggests the separation between population Y's ancestors and Oceanian populations occurred somewhere between 30,000 and 45,000 years ago, which is to say, well before the Beringia crossing. This timing is consistent with the idea that population Y descended from an early dispersal of modern humans out of Africa that moved through South Asia and into Southeast Asia before the ancestors of most East Asians had differentiated. They were, in a sense, an older layer of human expansion, the descendants of a migration wave that preceded the Northeast Asian populations, who became most Native Americans. How they reached South America from Southeast Asia or Oceania, crossing either the Pacific Ocean or moving through a northern route, no one has yet confirmed, remains the central unresolved question. The genome says they arrived. It does not yet say how. But here is where it gets complicated. The complication concerns what the discovery of population Y means for other ancient American remains, whose morphology has puzzled researchers. Kennewick Man, the 9,000-year-old skeleton found on the bank of the Columbia River in Washington state, was the subject of years of legal and scientific dispute. His skull morphology differed from modern Native American populations, leading some researchers to claim he was not ancestrally related to them. Ancient DNA analysis, published in 2015 by a team at the University of Copenhagen, found that Kennewick Man was most closely related to modern Native American populations, specifically to the Colville tribe of the Pacific Northwest. His unusual morphology reflected normal variation within an ancient population whose skeletal features had not yet fully converged on the modern Native American form. But population Y complicates even this resolution. If two distinct ancestral populations entered the Americas, and if their descendants interbred to varying degrees in different regions, then skeletal morphology from the early Americas may be a mixed signal. Some individuals expressing more of the population Y ancestral form, others expressing more of the mainstream founding population. The cranial variation that seemed to require a separate origin might, in some cases, genuinely reflect it. The deeper complication is about how we define Native American. If multiple founding populations entered the Americas independently, carrying ancestry from different parts of the Old World, then Native American ancestry is not a single stream, but a braided river. Multiple sources, joining and diverging across thousands of years of isolation. The population Y signal is weakest in North America and strongest in remote Amazonian and central Brazilian groups. Something happened in South America that did not happen in the north. Either population Y arrived in the south and spread northward only partially, or they arrived through a southern Pacific route, entirely distinct from the Beringia corridor, never passing through the regions that would become North America at all. That question has no answer yet. The genomes have opened it, they have not closed it. There is something almost mythological about this finding, though mythology is probably the wrong frame for it. Deep in the Amazon, in communities that had never read a paper on ancient DNA and had their own complete and sophisticated accounts of where their people came from, there survives a genomic record of a migration so ancient and so remote that it left almost no other trace. These are not historical people. They are not archaeologically visible. They left no Clovis points, and no rock art and no hearths that carbon-14 can date. They left only the encrypted record of their existence, buried in the cells of their distant descendants, waiting for a sequencing machine built 11,000 years after their time to read it. The Americas were settled twice. The second wave arrived undetected, not because it was hidden, but because we did not yet have the tools to look. Now we do. If you want to follow where this research leads next, because the question of how population Y reached South America is one of the most active puzzles in ancient human genetics, and new data is emerging every year. Subscribe. I cover every major study as it publishes. The rewriting of American prehistory is nowhere near finished. And the question I find myself sitting with, what other migrations are still hidden in living genomes, waiting for the right analysis to find them? The Americas were not unique. Every populated continent was reached by multiple waves of people across deep time, most of them leaving no written record and minimal archaeology. If the genomes of Amazonian tribal communities could hide a founding population that reached South America from Southeast Asia, what else might be encoded in the DNA of people living in places that history has assumed were settled by a single, simple migration? The past is not behind us. It is in our cells. We are only beginning to read it. Population. Why reach South America before the populations, whose descendants are the vast majority of living indigenous Americans? They were absorbed, demographically overwhelmed, their cultural contributions erased by every subsequent layer of history. Only the genomic shadow remains. It is haunting precisely because it is all that does remain. A faint signal in a sequencing run, preserved by biology when everything else was lost. Their names are gone. Their language is gone. Their stories are gone, but the cells of their living descendants remember them, encoded in a chemical alphabet that no conquest and no time has yet managed to erase entirely. The most pressing unanswered question in population Y research is not whether they existed. The genomic signal is too clear for reasonable doubt. But when and how they arrived. The leading hypothesis, proposed by Pontus Skoglund and David Reich in their 2015 study, is that population Y represents an early coastal migration along the Pacific Rim, following the same route that is increasingly proposed for the initial peopling of the Americas. Rather than crossing Beringia and dispersing southward through the continental interior, population Y's ancestors may have followed the Pacific coast, moving from Siberia through Alaska, down the western coast of North America, and eventually reaching South America along a route that hugged the kelp forest ecosystem, providing reliable marine food sources. This coastal route hypothesis, sometimes called the kelp highway model, is now supported by multiple independent lines of evidence, including the genetic data from Alaska and the dating of pre-Clovis sites. The Anzac child burial in Montana, dated to approximately 12,600 years ago, and associated with Clovis tools, carries none of the population Y signal, but coastal pre-Clovis sites in Chile, dated to 15,000 years ago, may reflect exactly the coastal migration that population Y was part of. The genomic evidence and the archaeological evidence are beginning to align.